It is a privilege to be here in this forum and a part of this good engineering practice document. As Priya mentioned, this was released in July this year, and it was created by a team of engineers from quite a few leading members of IPA. Could you go to the next slide, please? We did this document in two phases. Yeah, I was leading it, and there's colleagues from Lupin, Cipla, Torrent, and Jidus and DRL worked with us. Thanks to Prasanji and Rajendra ji for mentoring us in this journey. It was an interesting discussion and collaboration to create this document. Yeah, next. So this document, is a very comprehensive document on clean room design and requirement. So it contains a lot of topics which is not been, very specifically elaborated in many of the guidelines. So we have, created this document with collecting a lot of information from different guidance documents, engineering documents, and best practices, engineering best practices documents. The link is available here. I request all of you to download this document, go through it. I'm very hopeful it will be useful for all, while creating a new greenfield project, as well as while upgrading, modifying the existing project, buildings, facilities. This document will be very useful. Next, please. So it contains different chapters. So as mentioned here, it talks about clean room design for both for sterile facility as well as for solid oral facilities, and talks extensively about HVAC design on OSD plants, HVAC design in sterile plants. I think this is the only document which talks about detail about how PMS should be in pharma plants. So there are a lot of ambiguity and a lot of, because BMS is a general engineering solution being used by a lot of modern buildings. But whereas our pharma requirements are, bit stringent and different, so it needs certain level of attention. So we tried here to give a detailed brief how the BMS needs to be configured, designed and implemented in pharma plants. Then it talks about water system, which is again a very critical utility system for pharma plants. Then we talk about compressed air system and nitrogen system, how it needs to be maintained. Then in this document, we spoke about handling of documents and especially drawings. So like all engineers manage drawing in engineering colleges and institutes. How the drawings to be created and maintained is being taught, but pharma has a different expectation as far as traceability and requirements are concerned. So in past, we found there are citation in different companies related to maintaining engineering drawings. So we have kept a chapter here, how to create and maintain engineering drawings. Then it comes to equipment life cycle. The recent part, again, managing the equipments throughout the life cycle is becoming a challenge because the technology is getting upgraded rapidly, and while maintaining the equipment, maintaining it over life cycle has become very critical and crucial. So this guide specifically talk about it. Then comes the maintenance strategy. So maintenance is a very critical process in pharma plants, and what are the different practices and system needs to be followed. This guide talks about it. And at the last, it talks about the Gemba. Gemba, though there are independent guidance on Gemba, but since most of the Gemba part covers the facility and equipment majorly, so that's how we have covered some part in this guidance. Next. So in the next couple of slides, I'll specifically talk broadly on high level what are the things being covered in each chapter. So for the interest of time, then I'll stop and look forward for the questions. And when anybody will go detail in the download the document and go through it, there are a lot of information. I'm sure it will be useful. So in the guidance and clean room design requirements, so we are talking about facility design approach, design consideration, like what are the finishes required, what are the different grade and different classification area, what type of finishing is required. Specifically for solid whorls, what are the details or attentions are required, how utility needs to be managed in the facility. We are talking about it. We are talking about control system and instrumentation also. And in this document, we are talking about for sterile facilities, the various technology and isolators also being described in detail. Next. Then comes to the next chapter where we are talking about HVAC design for the OST. So as we all know, HVAC system is a very critical system for cross-contamination point of view and for environmental monitoring point of view. And both the facility needs different attention starting from the design, implementation, commissioning, and maintenance of HVAC system. So that's the reason we have kept two different chapter, one for solid oral plants and one for sterile plants. So in these, we are talking about like what are the different systems being used, how it being operated, how it is being designed, what are the design consideration needs to be uh, taken care during initial stage and how it needs to be maintained. Next, please. So then what are the different critical things which needs to be taken care during validation, operation, and how the maintenance needs to be done if there are certain because there are a lot of configuration available in the market, like it can be used at a different way, but cost of operation is another thing which we need to look at. And as we all know, the second highest operational expenses for a typical pharma plant is coming from the utilities, and HVAC is the 60% of the utility of a typical formulation plant. So looking at that, it become very crucial how we design our HVAC and how we operate and maintain. So that those parts also address in this document in detail. Next. So similarly, we have a separate chapter, as I told you, for a sterile plant, HVAC design for the sterile plant being described here. So where the solid oral design is bit different than sterile plants. Here we have to consider even the contamination, sterility, and continuous operations part into a very critical level. And the filtrations and classifications are, as we all know, are very critical and stringent in case of sterile. So accordingly, these chapters talks about room pressurization, number of air changes requirement, and zoning and leakages through doors, because room pressurization will highly depend on the leakage rate and all. So all these critical parameters for HVAC being described in these chapters. Next. Then it comes to the next chapter talks about the BMS system in the pharma. As I told you, the BMS is a very generic, building management system being implemented in all modern and new buildings across in even in commercial buildings, in other manufacturing setups, as well as in pharma. But looking at our requirement on validation and data management. and the process criticality, it becomes very crucial to design and control BMS system appropriately so that it can be, help effective and helpful during the long run. There are a lot of facilities in past couple of decades where the BMS are being installed but never been could not be validated and used due to accurate or correct consideration during initial stage, because the vendors who provide building management system today uh, in industry, they also provide the building management system to rest of the buildings, commercial buildings. And 90%, 95% of their business comes from there, so they used to implement in a way which is very generic and common without any validation approach and typical control systems which are required for pharma. So therefore it we need to uh, consider the choose the vendor correctly, and at the start of the URS, we need to specify our requirements and uh, design the system so that it will be really helpful and useful for long run. So in this chapter of the best practice, we talk about all those nitty-gritty of design of our plants, BMS plants. Next. So here comes it's this a little bit detail like how the BMS and EMS control systems are being designed. So because these control systems and configurations are crucial for, of course, validation and further operation and maintenance of the system. Next, please. In this chapter, it talks. So we talk about like what are the different type of hardware needs to be considered, how the networks control device needs to be chosen, the server database, what are the different types of application softwares are available, what is like the typical requirement of power failure, recovery, time synchronization. So these are the specific requirement which our industry needs, whereas vendor may not be aware because they are implementing this system while implementing this system in the commercial building. So alarm management and warning. Now the current expectation on alarm trend, which is coming from the regulatory, which has come as a regulatory requirement, which is also very critical. So all these topics are being discussed in the best practices of this chapter in detail, and which I'm sure will be useful for any new system design and configuration. Next. Similarly, coming to the water system, so water is another, next to HVAC, the water is another very critical utility for manufacturing. When it comes to injectable, it even become like a raw material for us. So in this document, we have starting from the potable water till the water for injection, all these steps are being described, the requirements are being described, what are the digital design criteria needs to be considered, what are the key factors which needs to be considered is being discussed in this document in detail. Next, please. So these are the different components or systems of pharmaceutical water system which are being discussed in this document in detail. Next, please. Next utility which plays an significant role is compressed air and nitrogen in the process. Here, starting from the design selection, treatment, air treatment, then filtration, and how the whole network is maintained is being described here. So as we all know, the moisture content in the compressor becomes very crucial, and while designing the system, uh, considering the moisture content and the array, having the appropriate system to address the moisture in the compressed air is crucial. So all these and the filtration of the compressed air. So a lot of times there are places where we do we have observed gaps in compressor filtration when it comes to products coming in contact or products sterile products being manufactured. So appropriate filters for the both for nitrogen and compressed air needs to be chosen during project design stage, which has been defined in this document uh, in detail. Next. So that, as I told in the initial description, so we have covered a chapter on the handling of drawings, engineering drawings. It's specific to pharmaceuticals. So all engineers, while in their colleges or school, being taught about how the engineering drawings needs to be drafted or managed. But when it comes to our industry, the pharmaceutical industry, the regulatory point of view, managing this document as per the good documentation practice, so become a very crucial process. And as I told in past, there are quite few instances where there are citations, regulatory citations, because of drawing management, how the drawings are being updated, managed, and numbered, and approved and kept. So therefore, we were advised and guided by our mentor to cover this chapter, and I'm very hopeful this will be useful for all of us. As a reference, again, this type of guidance, I don't think any of the major guideline covers them, but these are the good best practices which needs to be followed, which will be definitely helpful. Next. Then we have a chapter where we talk about the life cycle management of the equipment. So in the recent past, there are a lot of regulatory citation again on equipment conditions, and so many a times we install, validate, qualify the equipment, and then we keep on maintaining and using them. Most of the time we didn't have a process how it will get retired or assessed for its fitment. So we keep on requalifying till it may fail on qualification. But this document extensively describe how it needs to be managed over the whole life cycle of the equipment. So this chapter again will be very helpful for aged facility as well as for new facility to implement this system, so that it will take care of the quality aspect of equipment and facility on a long run. Next. Uh, So these are the different steps, what is being described, starting from the replacement, life cycle documentation, different stage of qualification, preventive maintenance, and importance of AMCs. So these are the things being covered in this guidance document in this in this chapter. Next, please. The next comes is very critical aspect of maintenance. So there are different types of maintenance, and how it needs to be planned and implemented in uh, our typical pharma manufacturing environment being described in this document, and starting from TPM, schedule maintenance, on schedule maintenance, then reliability centered maintenance, then how it needs to be planned, how what are the different approach being followed in industry as well as different pharma or big pharmas, then how to validate the effectiveness of maintenance. Like many a times we keep doing maintenance, but how to evaluate our maintenance program is effective, like measuring the MTBF, MTTR. So all these detailed aspect of maintenance strategy being described in this chapter. Next. Next is the Gemba. So I think all of us know about it. There is a dedicated guidance from ISP on this, and this being practiced by all, I think almost all companies today. But since it is when we go for a Gemba, it covers majority part of facility and equipment. So we thought of covering in this guidance as well. So I think it's quite similar to all what we are practicing. Next. So these are the different references we have taken while drafting this document, starting from US FDA, EU, WHO, and PICS guides. So that's all about this best practice on facility and equipment. And uh, I'll urge everybody to download this. It is freely available in IPA website, and go through it and implement those practices whenever needed. Thank you.
IPA best practice document, PAD tools in oral solids and APA. It is published in IPA website, and one can go through that. And the slide is just a brief, very brief of the guideline. Next slide, please. These are the authors, and thanks to the organization like Sun Pharma, Cipla, Torrent, ZS, Dr. Reddy's. Next slide. PAT is part of QBD. This we can understand from the definition given below the diagram. There are two definitions. One is QBD, defined by ICH. The other one is PAT tool, defined by US FDA. And if you see this definition, one common goal is mentioned: how to get the consistent predefined quality that is mentioned. So by this is obtained by scientific understanding of the process, product, then quality risk assessment. This is the way to achieve the consistent quality, and this is explained in QBD as well as PAT guideline by FDA. So that is why PAT is part of QBD. Next slide. We can see here the diagrammatic explanation of the variable input resulting in variable output quality. There is a variation in the CMA, critical material attribute of raw materials, which can vary from, let's say, for example, high moisture content in one lot of lactose and smaller particle size in other lot of lactose. So particle size may vary or moisture may vary. Because of particle size variation or moisture variation, the blending time may not be similar. It can also vary. So that's why variable output quality we get. With the help of PAT tool, we can go for variable process. It is permitted by the regulatory agency also. And when we go with variable input raw materials, we can get always fixed quality output. That's why FDA has said that the time-defined or clock-based endpoint will result in failure because of the physical variation in the raw materials. Physical variation means moisture content and particle size and all. And your desired endpoint should be, it is not clock-based one, it should be the product quality-based one. Next slide, please. So here we can see modes of measurement. Although we know what is offline, at-line, online, but we need a regulatory definition, what is online, at-line, and in-line. Otherwise, it looks like all are similar. Okay, so if you see briefly what is online, in-line. So in the measurement, the sample is measured, in case sample is measured in the mainstream of manufacturing, then it is called inline. If it is sample is diverted towards the parallel stream and measured, then it is online. And here the sample is always diverted back to the mainstream. In case of outline, the sample is measured near the manufacturing machine or manufacturing place, and the sample may or may not be returned after the measurement. And in case of offline, the sample is measured in QC or any other place by shipment of sample from one place to other place, and it never get returned after analysis. Maybe it is a destructive analysis. Next slide, please. So these are the PAT tools available at different stages, starting from API manufacturing to till coating. So we can see the critical parameters which is to be monitored for each manufacturing stages, and then corresponding available pad tools also. Next slide. This is very important implementation procedure. We need to form a pad tool team comprising of formulation department, manufacturing, then QC, QA, regulatory affairs, and statistician, of course. Select a product basis the failure history and commercial volume. So we should not only look for failure history, and we should not select a low volume, low commercial volume product. We cannot justify the rate of the ROI of the instrument. So that's why we should select a high volume commercial product with failure history. And also we should see impact in the existing process or approval. Suppose your product is a complex product, and then if you do a variation filing with the PAT tool, and a regulatory agency may demand for some additional data like discriminatory dissolution data, many other things. Then correlate with, hello, yeah, correlate with, please, previous slide, please. Yeah, previous slide, please. Yeah, correlate the process control with the CQA, applying statistics, then identify a pad tool based on the monitoring parameters, accuracy, prerequisite, MOC. MOC, why? Because some probe may be in contact with the sample. In that case, we need to have a stainless steel pharma grade, which is applicable for the probe, which is in contact with the direct contact with the product. Installation of pad can be done under the facility's own quality system. There is no need of regulatory approval. But however, the implementation can be done after approval by the regulatory agency. A study protocol may be prepared, and we need not communicate to the agency, but if necessary, we can communicate and get the confidence. Model development trials are taken, and then model validation. Then supplementary filing can be done with CB30 or PASS route, and it should be submitted for getting approval before implementation. These are the brief view, although it is in detail, it is given in the guideline. Next slide, please. Challenges in implementation. Why your company cannot implement, although a lot of details available in the internet or book. There is a lack of experience, own experience and expertise in selection of tools, method development, validation, and interpretation of the results. And then burden of investment. Already cost reduction program may be going on, and this PAT tool may be an additional burden. Time limits for the new development. Some complex product has same time limit as that of the simple product. Then hesitation to change an approved process, as it may require an approval again with more data as of today's guideline, like discriminatory dissolution, and then elementary risk assessment, etc. So risk in data generation from commercial batches due to SOP, GMP, and regulatory audits. PAT is pet projects in some cases, like pet project for the manager or director level with no resource or low resource and least priority in the product list. And possible solution can be a quantified business case. We can have a cost and benefit analysis for instrument manpower, which we spend, and that also benefit we get from batch, saving the batch, avoiding the batch failure, and then time saving. Then sponsorship we should get from the top management, and FDA doesn't audit the data generated in the manufacturing site for research purpose. That is clearly mentioned in the FDA guideline. So we hope the same from the other regulatory agency also. Next slide, please. So here is one example, NIR spectroscopy as the inline tool. So we can see in the picture that the first picture is showing the traditional sampling using sampling thief, where there can be a segregation due to sampling and then transit of sample from one lab to another lab, HPLC analysis, all this. So this can be avoided by using NIR tool, which is attached to the V blender, and it is continuously monitoring live and no need to sample and wait for the analytical data. And so the content uniformity testing, the picture shown adjacent to this. Next slide, please. So here is the bunch of spectra, NIR spectra, which shows the overlap with each other at the bottom, whereas it is not that much similar at the top. That is because the spectrum in the top is 2 minutes observation, and at the bottom it is 30 minutes observation. That's why when the time progresses or blending rotation increases, the uniformity increases. That's why if you can see in the adjacent picture, the RSD or variance is decreasing and reaches a minimum value and gets stabilized to find an endpoint. Next slide, please. So this is typical calibration model. Based on the R square value or slope value, we can increase the number of trials to strengthen the model or decide it is enough. Yeah, please, next slide. We can start the experiments for model building purpose, starting from 70% or 75% up to 130 or 125, depending upon the possible variation in the assay value or content informative value. So take these trials and then analyze the NIR, then HPLC, and correlate it as shown in the previous slide. We can plot a calibration curve. Next slide. Next slide, please. Model building exercise can be done. This is complete by this way. Like we can take a decision basis, this flow chart, and it is given in detail in the guideline. Next slide, please. Yeah, PAT tools for high shear granulation. This is another example for the next unit operation. So available choices for granulation is ammeter reading, indirect Turk method, then drag flow force sensor, impeller Turk sensor, then comes near infrared sensor, then thermal effusivity sensor. So depending upon the, they have its own advantage and disadvantage. For example, ammeter has less sensitivity because it measures the entire motor load, not just only the impeller load. And the indirect Turk sensor method is like it is just calculating from the ammeter reading, so the sensitivity is again less. Drag flow sensor is more sensitive, but it can be imported from one RMG to another RMG. That is an advantage, but it is actually not scale independent. Then comes impeller Turk sensor. It is permanently fixed, so we cannot port it from one RMG to another mixer granulator. And it is more sensitive method compared to the ammeter reading. And near infrared spectra, this sensor is more suitable for aqueous process, although it can be used for other process also. Thermal sensitivity sensor, it is scale independent. That is one advantage and preferred in the aqueous process. So it has, every sensor has some its own plus and minus depending upon the product and process we can select it. Next slide. Since it is new, we have shown here this sensor is new in the market, drag flow force sensor. It is coming directly in contact with the powder. And when powder is rotating, powder is moving in the RMG, it gives some drag force towards the needle type probe. So the drag force is plotted in the y-axis against the time in the x-axis. When dry mixing happens, the drag force is less. And when fluid is added, because of the strengthening of the densification of the powder, the force is increasing, and then it reaches a maximum. Then after chopper action predominates, the particle size decreases, lumps are broken, then force is coming down and gets saturated. So this is a signature we can call process signature. We need to match every commercial batch compared to the reference batch, so that process signature is matched and endpoint is reached to get the consistent product quality. Next slide, please. So this is about the drag flow sensors, whatever I explained in the previous slide, that is given in the line by line, and in detail we can get the guideline. And 10 or more batches are required to show overlapped signature profile to build a model and get the get the consistent output quality. Then audit trials should be available for inspection or audit. No need of approval from regulatory agency for additional data generation, only for implementing the flexible fluid uptake, flexible kneading time, we should get approval. And once we get approval, we can change the kneading time every batch according to the raw material moisture or particle size. That is the beauty of this pattern. Next slide. This is other variant. This is Turk sensor, which is installed near the impeller. It measures exactly the load in the impeller, not the entire motor. And it measures the rotational force in the impeller. And the rotational force is resisted by the blend when the blend gets more weight, it gives more resistance, and accordingly the Turk value is plotted. So we can see in the next slide a typical Turk profile. The Turk profile increases when fluid is added in the granulation because of the densification. Then suddenly it comes down because of chopper action, and again it goes up because of the secondary granule growth and gets saturated. So this is your end point. Next slide, please. This is typical graph. So here is one variation plot, like one person has opened the lid. The first slide is the right one, the reference batch. The second one is variation batch. The operator has opened the lid and then manually de-lumped the granules and then again closed the lid, restarted the process. So the endpoint is disturbed here. We cannot match the reference batch here. So this can be there when we intervene manually. Next slide. So here it is numerical value compared, like this is a case study batch. So we can see amperage value for each stage. Initially dry mixing, then water is added, kneading 1. So granulation endpoint is not reached, then kneading 2. So between kneading 1 and kneading 2, there is no significant difference in amperage, but there is a significant difference in the Turk value. So that's why Turk sensor is more sensitive. We have seen in the previous slide some tabulated form. Amperage reading is less sensitive and Turk sensor is more sensitive because it is directly measuring the amperage impeller load, not the motor load. And batch 1 and batch 2 are reproduced. Similarly, 10 batches we can have with all input variables, moisture and particle size, and vendor change, raw material vendor change. All these we can do, and model building we can do. Finally, we file the product and then go for the endpoint based granulation instead of clock based time based granulation. Next slide. So this is for fluid bed process coating. This is related to direct image analyzer. Direct image analyzer is attached to the Worcester. When the pellets are fluorescing, it is illuminating, the image analyzer is illuminating using the LED light each particles and then capture in the camera by distinguishing the overlapping particles and plots the particle size growth with the time as shown in the adjacent figure graphical form. The growth of particle happens and the signature has to be overlapped between 2 commercial batches or 3 commercial batches, and we can build a model and then always further subsequent batches should match this one. And the advantage is we have experience like for unloading and reloading the pellets in multiple stages of coating. It takes a lot of time in the manufacturing area. In R&D, it takes very less time, but in the manufacturing setup, it takes a lot of time for unloading and then checking the weight, then reload it in case of multiple stages of coating, seal coating, functional coating like this. We can have a final check after functional coating, but in between multiple coatings will be the drug layering and seal coating will be there. In that case, we don't have to unload it and then check the weight of the pellets to see what is the coating weight gain. We can always correlate with initial 10 batches, what is the coating weight gain and what is the corresponding particle size. So corresponding particle size is live plotted by this instrument in the computer. So basis that we can decide whether go for the next stage of coating or continue the first coating. No need of unloading and reloading. Next slide, please. So by this way, it saves the time and money, and we can justify this way for implementation. And this is fluid bed granulation. Previously, we saw fluid bed coating. This is fluid bed granulation inline PSD tool. Here, this is a typical diagram, and it is clearly explained in the guideline. So in short, we can say here, we are charging the powder, and then spray is started. Once spray is started, the granule grows, and then it reaches maximum. And when drying happens, due to filter back shaking, the fines are generated and the particle size is coming down. This is a live profile of PSD inline data, and we have to overlap this reference batch for subsequent commercial batches. Next slide. The final one, this is the pad tool for APA crystallization. This is FBRP in crystallization. It measures the particle size by introducing the probe directly into the sample during crystallization, and the chord length distribution gives the information about the particle size. The detailed information is given in the guideline, and you can ask your questions. in the interest of time.